Okay, so like I said earlier, now that your smear has been prepared, you are now ready to start the staining procedure. The first stain that you will be doing is the gram stain. Now this stain enables the user to examine the bacteria and determine whether bacterium in the sample is gram positive or gram negative. And like I said before you start any experiment, make sure that your space has been disinfected with germicide and wiped down with paper towels. And when you're done with the paper towels, just put them in the biohazardous waste container. But since we're continuing on with the experiment, also make sure that you have a new pair of clean gloves on and that you have your lab coat and a pair of goggles on, because with this staining procedure, things can splatter, so just want to keep ourselves safe. And then, of course, you would, if you were in a real lab, you would go and gather your materials, which I already have set out right now. So, of course, you have your heat fix slides, which are in the slide box right now. I know you can't see it, but there it is. And then you need a stain tray. I don't actually have one, so we're just going to make do. Your slide clip. Sorry, you couldn't really see that from where it was. Then you would have your crystal violet stain. Yes, I am using paint bottles to make it look like it's those. Grams iodine stain. Oh, before I forget, the methylene blue stain is going to be your primary stain. The grams iodine stain will act as your mordant. This will help the bacterial cells in your smear to retain the crystal violet stain better. So that would be those gram-positive bacterial cells that would retain the crystal violet stain much better. This is your 95% ethanol. This will be your decolorizer for this experiment. Whoops. This is your safranin stain. This will act as your counter stain. You are also going to need a water bottle for rinsing. I don't have the actual nice one that they have in labs, but just as a thing, we're gonna pretend that that is what it is. Babyless paper, stain collection vessel. I do not have one of those. A slide box, biohazardous waste. To start this experiment, you're gonna take your heat fix slide that you made earlier, this guy right here, and put it on the stain tray. Now that the slide is on the stain tray, you can apply your first stain, which is your crystal violet. Like I said earlier, this is your primary stain. So you're going to apply this to the stain for about 30 seconds. Now I'm just going to speed this up a little bit, but once you've applied that stain, let the slide sit for about 1 minute so that the stain can seep into the smear. Now what you're going to do after your slide has sat for 1 minute is you're going to take your slide clip, pick up the slide, and you're going to rinse it off in a gentle stream of water. Make sure to tip the slide downwards over the stain tray and just gently rinse the slide with water to get that excess crystal violet stain off. Then put it back down. Okay, so the next step that you're going to do is you're going to flood your smear with grams iodine. And you're going to apply this to the stain for about 60 seconds. But for this experiment, I'm just going to speed it up. And once you're done doing that, you're going to pick up your slide clip, use it to grab onto the slide, and like you did earlier, you're going to tip it down and you're going to rinse the slide with water gently, just to get that excess iodine off. Now, while still holding your slide, you're going to apply 10 to 15 drops of your 95% ethanol to your slide. This is going to act as your decolorizer. Make sure that you don't apply the ethanol directly to the smear. Just allow it to run down it. And once you've put drops there, you can put that down. And while you're still holding the slide, You're going to rinse it off in a gentle stream of water to get that excess ethanol off. Once you've done that, put the slide back on the stain tray. The next and final stain that you'll be using for this part of the experiment is safranin. Now this you're going to apply to your smear for about 30 to 45 seconds. Now the safranin stain acts as your counter stain. Unlike the crystal violet stain, which turns gram-positive cells purple, this stain, safranin, turns gram-negative cells a pinkish reddish color. So I'm going to speed this up a little bit. After you've applied the safranin for 35 seconds, put that back down. And then what you're going to do is take your slide clip, grab the slide up, tilt it downwards, and rinse the slide off with water gently. You want to get that excess safranin off. Once you've done that, put it back down on the stain tray. Now that all the stains have been applied to the smear and it has been rinsed off, you can now blot dry the slide using your book of bibulous paper. So what you're going to do is you're going to put it in gently and just gently rub the slide in to dry it off. Put that down, and then once you're done, just put your slide in the slide box for safekeeping. Now, since in this experiment you would have two heat-fixed slides, I know I don't have the second one, but essentially you would repeat the steps all over again from where you applied the crystal violet, the Graham's iodine, the ethanol, and the safranin to it, rinsed it all off, and then dried it, and then you would have that second slide as well. And then what you would do after both slides have been completely dried off using the blot paper or blotting paper, you would view the slides under the microscope. You would make sure you view them under the objective lens that has 1000x magnification so that you can actually see the specimens. Once you were done viewing the slides under the microscope, you would place the slides back in the slide box to keep them safe, which I already have. Just gonna close that. And then, of course, you would pour any liquids that you had in the stain tray from your experiment into the stain collection vessels for them to be disposed of. And then, of course, you would move all your materials out of the way and then disinfect your workspace again. You would use germicide to kill or inactivate any microorganisms that are present on your benchtop. And then you'd use paper towels to wipe up and dry off your workspace. Once you were done with the paper towels, just throw them away in the biohazardous waste container. And then once your workspace has been cleaned, you can remove your gloves and put them away in the biohazardous waste container as well. Then go and wash your hands, again, very thoroughly. And then once you've washed your hands, put on a new pair of gloves because you're about to start the next experiment, which is your acid-facet staining.